Hello students, I'm Dr. Arpita De. The topic for today's discussion is double fertilization in plants. So before we start our lecture, let's first know what is double fertilization. Double fertilization is a complex phenomenon in angiosperms or flowering plants where one sperm fertilizes the egg and the other sperm fuses with the two polar nuclei of the large central cell of the megagametophyte, thus giving rise to two things. What are they? Giving rise to a diploid zygote and a triploid nucleus respectively. So when one sperm, right, one haploid sperm fertilizes one haploid egg cell. One sperm is fertilizing the haploid egg cell. This gives rise to a 2N zygote. All right, this gives rise to a 2N zygote. This is known as syngamy. This is known as syngamy, right? Now, this one, what is going to happen to this? This becomes our endosperm. Look here, this is a diagrammatic representation of the embryo sac where you can see this is the 3N endosperm and this is the diploid zygote. Now, we'll learn a little more about these. See. The pollen grain adheres to the stigma of the carpel. This is the stigma. This is the stigma part. You know, stigma that it looks like this. This is the style. This is the stigma, right? The pollen grain comes and rests on the stigma. Thereafter, what happens? It absorbs water or moisture and then germination of pollen tube starts, and that pollen tube starts to travel or extend this downwards. Okay, after it extends downwards, it slowly goes to meet somewhere at this region. It goes to and penetrates this part. This is the micropilar end. Okay, and this is the chalazal end. Right, this is the micropilar end and this is the chalazal end. Fine. Now see, this pollen grain has absorbed moisture and the pollen tube germination has taken place over here. Pollen tube enters through the micropilar end. This is the micropilar end as I have just said, right? This is the micropilar end and the pollen tube enters through this and then it releases the two sperm in the megagametophyte. These are the two sperms which are going to be released into the megagametophyte. Okay. Now we will learn about the structure of a matured embryo sac where the arrangement of the cells in an unfertilized ovule will be in this order. Now let's see what is this 3, 2 and 3 all about. So let's draw a matured embryo sac. Okay. This will be the micropilar end. Micropilar end means which one? This is the micropilar end. This is the end from where the pollen tube penetrates. Fine. Now, here at the micropylar end, there will be 3 cells. I'll just use a separate color so that it becomes easier for you to understand, right? These cells are the synergids. These are the synergids. Okay. Next, right in the center, you will have the central cell. Okay. This is the central cell and these are the polar nuclei. Fine. These are the polar nuclei. And at this end, at the chalazal end, we will have again a set of 3 cells. These are the antipodal cells. What are they? These are the antipodals. Okay. These are the antipodals. Fine. Now, we have 2 synergids, this one and this one. Now, what is this one then? This is the egg cell. All right. This one is the egg cell. Fine. Now, the sperms will come inside and will fertilize this egg cell, the process known as syngamy, and the other sperm will fuse with these 2 polar nuclei and will form 3N endosperm. Is that clear? This will form endosperm. One will form endosperm and the other will form the diploid zygote. Fine. So, now did you understand this arrangement? 3 cells over here, that is 2 synergids, 1 egg cell, 2 polar nuclei, and 3 antipodals. Is that clear? Now let's move to the next part of our discussion. Now, the 2 central maternal nuclei or the polar nuclei that contribute to the endosperm, these they arise by mitosis from the single meiotic product that gave rise to the egg. Okay, the maternal contribution to the genetic constitution of the triploid endosperm is double that of the embryo. All right. The large cell of the gametophyte will then develop into the endosperm. Now, what is this endosperm? What is the function of this particular tissue? Why is it so important? It is important because endosperm is a nutrient-rich tissue that provides nourishment to the developing embryo. That is the reason why endosperm is so very important. Okay. So this is how we come to the end of our discussion. Let's give you a short take-home message. Usually, this double fertilization phenomenon is observed in angiosperms or flowering plants, but this is also observed in another set of non-flowering plants in the order of Nitalis. So, double fertilization also occurs in non-flowering plants in the order Nitalis other than angiosperms. I hope the discussion is clear to you. If you have any confusion anywhere, please feel free to contact me. Get back to me. I'll try my best to solve your doubts. Thank you.